Welcome to this explainer. Today, we're diving straight into one of the most intricately designed systems known to science. We're heading inside the eukaryotic cell, but we're not just going to list off dry biology terms. Instead, we're going to look at the cell for what it truly is, a highly coordinated, insanely efficient biological factory. Specifically, we're going to explore the endomembrane system.
So think about this for a second. How does a microscopic cell manage an industrial-level supply chain with such flawless efficiency? Literally right now, your body is juggling a massive number of tasks simultaneously. It's digesting food, it's firing electrical messages through your nerves, pumping blood, filtering waste. And all of that is only possible because every single microscopic cell is equipped with its own heavy machinery, which we call organelles. And just like a sprawling industrial complex, this microscopic machinery has to work together perfectly.
To figure out how the cell pulls off this logistical miracle, we really need to understand the endomembrane system. Basically, this is a group of membrane-bound organelles inside eukaryotic cells. But the absolute key here is that word coordinated. These organelles aren't just floating around randomly. They form a literal supply chain. Their functions are deeply intertwined to synthesize proteins and lipids, process them, package them, and transport them to their exact destinations. Endo literally means within, so this is the cell's ultimate internal logistics network.
This interconnected cellular network boils down to 4 core departments. Even though a cell has tons of structures, this exclusive endomembrane club consists of just these 4 players: the endoplasmic reticulum, the Golgi complex, lysosomes, and vacuoles. Working together, they handle the entire life cycle of cellular products.
All right, let's head to Section 1, the manufacturing floor. We're going inside the endoplasmic reticulum, or the ER for short.
You can picture the ER as this massive, sprawling network of tubular assembly lines where the cell's raw materials get produced. It's this incredibly dynamic structure made of membrane-enclosed tubules and flattened sacs, which are called cisternae, and it sits strategically right next to the nucleus. Since the nucleus is basically the corporate headquarters of the cell, the ER is perfectly positioned to receive genetic instructions quickly and just get straight to work manufacturing what the body needs. And right there on the factory floor, there is a very strict division of labor. The ER is actually split into 2 distinct manufacturing zones. First, you've got the rough ER. It's called rough because it's studded with tiny, non-membranous structures called ribosomes, so it literally looks bumpy. Those ribosomes are the workers physically synthesizing proteins. On the flip side, we have the smooth ER. No ribosomes here, so it looks perfectly smooth. And instead of proteins, the smooth ER is entirely dedicated to synthesizing lipids, lipoproteins, and glycogen. So it's 2 highly specialized zones making up one incredibly efficient manufacturing floor.
Now, once those biological products are manufactured, they can't just pile up. They need processing and shipping, which brings us to Section 2, the Golgi apparatus.
I absolutely love this because biological texts explicitly describe the Golgi apparatus as the cell's receiving and shipping department, which completely validates our whole factory metaphor. Composed of these flat, disc-shaped membranous sacs, its primary job is to take the raw materials generated by the ER, package them up, and prep them for secretion.
Let's track the actual logistics flow here, because it's a literal, physical journey across the cell. Step 1. The product is synthesized over in the ER. Step 2. A small piece of the ER membrane actually pinches off, creating a tiny biological transport truck known as a vesicle. In Step 3, this transport vesicle travels to the Golgi apparatus and fuses with its receiving dock, which is called the cis or forming phase. The materials then get modified as they move through the Golgi. Finally, in Step 4, the fully packaged, processed vesicles pinch off from the shipping dock, the trans or maturing phase of the Golgi, and they're completely ready for delivery.
So where do these biological delivery trucks actually end up? Well, once they leave the Golgi's transphase, they basically have 2 main destinies. They either get delivered to local intracellular targets, meaning they stay inside the cell to support other departments, or they travel all the way to the cell's outer boundary, fuse with the plasma membrane, and secrete their contents completely out into the body. This right here is exactly how your cells release necessary hormones, enzymes, and all sorts of vital substances.
Of course, any efficient factory is going to generate waste, and it's going to need robust storage space. That brings us to Section 3, lysosomes and vacuoles.
Let's take a look at lysosomes first. These are essentially the specialized recycling and waste management centers of the cell. These single membrane structures carry a legitimately dangerous payload, extremely powerful hydrolytic enzymes. And these enzymes are capable of digesting all types of macromolecules.
Whether the cell needs to clear out old, broken-down parts or digest large nutrient molecules, the lysosomes break them down safely inside their membranous walls so the rest of the cell doesn't get damaged or cluttered up.
Meanwhile, we've got the vacuoles. You can think of these as the essential storage warehouses. They are membrane-bound organelles that hold on to vital materials, things like water, nutrients, or even waste products before they're processed. In plant cells especially, vacuoles play a massive role by providing actual physical structural support to the cell, keeping it rigid and healthy. So, working hand-in-hand with lysosomes, they keep the cell's internal environment super clean, organized, and well-stocked.
So we've covered the 4 main departments, but what's equally fascinating is Section 4, the excluded organelles. Let's talk about who exactly doesn't make the cut for this exclusive cellular club.
Think about some of the most famous non-endomembrane organelles. You've got mitochondrium, universally famous for being the powerhouses of the cell that generate ATP energy. You've got chloroplasts, those vital photosynthesis engines in plants, and peroxisomes, which act as microbodies handling intense oxidation reactions. All 3 of these are absolutely critical to the cell's survival. And yet, despite being membrane-bound organelles, they are firmly left off the endomembrane roster.
So, why the exclusion principle? Why are they left out? It really comes down to this. They operate basically as independent contractors. Their specific functions, like generating power or conducting photosynthesis, just aren't coordinated with that core synthesis-to-shipping supply chain we just walked through. Because they don't directly participate in that sequential relay of producing, packaging, and transporting proteins and lipids alongside the ER and the Golgi, they simply aren't part of the endomembrane system.
To wrap up this explainer, I really want to leave you with a thought. Can you even fathom the sheer scale of this microscopic supply chain? I mean, from the assembly lines of the ER to the shipping docks of the Golgi, right down to the hazardous waste management of the lysosomes. This perfect, non-stop efficiency is happening inside microscopic factories operating millions of times over, keeping you alive and functioning at this exact second. It literally redefines what we think of when we hear the words supply chain. Thanks so much for joining me on this journey into the cell.
Thanks for watching, friends. Don't forget to like and subscribe. See you in the next one.